All right, so today's topic is the human genome and gel electrophoresis. So a term that we need to be familiar with, and we've seen this time and time again, especially throughout genetics, is a gene. So this is a sequence of DNA that codes for a protein. So typically humans have about 25,000 genes. And these are located on our chromosomes. So each chromosome contains thousands of genes. Because we have how many chromosomes? 46, right? So the human genome is the entire collection of the 6 billion base pairs in each and every cell. So what if we were able to search all 6 billion A's, T's, C's, and G's in the human genome to identify disease-causing alleles? What do you think we might be able to do if we were able to do this? Okay, we might be able to cure all kinds of diseases. So in 2001, the Human Genome Project, also known as HGP, successfully sequenced the entire normal human genome. So by having this point of reference, scientists are able to identify defective genes that cause diseases. And we've seen and researched several different diseases, especially in our genetic disorder projects. Okay, we saw several diseases when we talked about karyotyping. We saw several yesterday in genes in the environment. Okay, so in theory, once defective genes are identified, then diseases can be cured. So the HGP led to several developments that have benefited humans. So now we're going to go through some genome sequencing. So if we look up here, this just shows us how this is possible. Okay, so here's our bear. We extract DNA from the tooth, and then we run it through DNA sequencing, and then we are able to compare DNA. So many diseases are caused by defective proteins. So in gene therapy, and this is a term we should be familiar with, an absent or faulty gene is replaced by a normal working gene. Okay, so think of it kind of like physical therapy. Why would you go to physical therapy? Okay, so if you had some type of surgery, right? Sure. You go back. You go to PT to strengthen, rebuild, right? So gene therapy, we take a non-functioning gene and we replace it with a normal functioning gene, or a gene that is faulty, and we replace it with something that is normal. So once we do this, the new functioning gene can now produce normal protein. Okay, so here's our cell with our non-functioning gene. We put in a functioning gene, and now the hope is that this cell will function normally. So a virus, okay, is modified so that it cannot cause disease. And then a DNA fragment containing the replacement gene is spliced to viral DNA. So we modify a virus so it doesn't cause the disease. And then we take a DNA fragment that contains the replacement gene, and we put it into the viral DNA. So we've got our DNA, We got a replacement gene, and then when we insert it in, this is what it looks like. The patient is now quote unquote infected with the virus, which injects the genes into the patient's cells. And then lastly, the patient starts producing the correct protein. Another term we should be familiar with is vector. So that virus that we actually inserted is known as a vector because it is what transmits the DNA to the cells. So for all the promise that it actually holds, in most cases, gene therapy remains still a very high-risk experimental procedure. So some of the risks, okay, the new gene may be inserted into the wrong location in the DNA. We can have an immune system reaction. The wrong type of cell can become infected. And another possibility is replacement protein can be overexpressed. Remember, we talked about expressing genes, turning them on and off. Okay, so up here is just a picture of a gentleman, okay, who died from the result of gene therapy in a clinical trial. So there are still lots of questions about gene therapy. So now comes the biggie. So gel electrophoresis, which we're going to talk about DNA fingerprinting. So DNA fingerprinting analyzes regions of DNA that are highly variable and they are unique to each individual, okay? So just like a normal fingerprint, there are no two fingerprints that are the same. So gel electrophoresis, which if we were able to do labs, we would be able to do this. Gel electrophoresis is a piece of equipment, it's technology that's used to visualize DNA fingerprints by separating strands of DNA by size. And the reasons we would use something like this are in order to identify crime scene suspects. We use it for paternity testing and also to show relatedness between different species. We're going to look at the top two today. All right, so the way this works. A small region of DNA is selected and it's cut with the restriction enzyme. So we need to remember this term, restriction enzyme. They cut. So remember, we talked about enzymes way back at the beginning of the semester. Who remembers what an enzyme is?
They lower the
Speeds up a chemical reaction, okay? So it is a catalyst, a protein catalyst that speeds up a chemical reaction. So restriction enzymes are used for cutting. So they cut DNA at specific sequences. Correct. Next, we use what's known as a pipette. to take the DNA and put it into different wells of a gel. Okay, so usually in this machine, this contraption piece of technology that we use, which looks like this at the bottom, okay, there is a gel that goes in the bottom, and then the pipette is what we use to take up the DNA, and then we insert that DNA into the different wells of the gel. And then we run an electric current through it, and then that's what separates the DNA. So here's where the electric current is applied, and it separates the DNA by size. So DNA is actually negatively charged, so it's going to move towards the positive charge, okay? So the smaller strands of DNA are going to move further and a lot more quickly than the longer strands. So think of it like this. If you were to have a 25-pound med ball, right? And a 100-pound med ball, which one are you picking up and getting further? The 25-pound one, right? 25 or 100? 25, right? Okay. It's lighter and you're going to be able to move it a lot more quickly. So you can compare this to that. So the smaller strands are going to move further and a lot more quickly. So if we were to look here, if these are our wells where we inserted our DNA, right? And the DNA strands are going to move across the gel, where are the smaller fragments going to be? The bottom, right? So another key, remember we said DNA is negative. Where do you see the positive? So this is where the DNA wants to go. So we're going to look through some examples because this is what you're going to need to know how to do. If you were to see DNA fingerprinting, you need to be able to figure out the suspect, the dads, okay, different scenarios like that. So DNA fingerprinting is used for crime scene investigations. So this just gives us an example. To the left is our crime scene sample, right? And then to the right, we have 3 different suspects. Answer's already up here. But what you want to do in order to figure out is match the bands. So this one is super easy because they look exactly alike. Does everybody see? Okay. Sometimes they're not as easy to figure out. Okay. They're not usually difficult, but some are a little bit harder than others. So if you were to look here and you were to look here, they're pretty easy to figure out because the banding patterns match. Does everybody see that? Okay. So here's another one. So here's our crime scene. And we've got suspect 1, 2, and 3. So who did it?
2.
Suspect 2. That's another one. They match. So now we got our DNA from the crime scene. This time we got 4 suspects. So who done it? 2. Okay. So these banding patterns are going to match up. And sometimes you can see them immediately, right? But in some cases, you might have to line up with a paper or a ruler.
There's so much like responsible to tell.
We're getting there. Okay, here's another one. So we've got semen that was recovered from a victim, the victim, the accused, and an unrelated person. Okay?
The accused. The accused. It's not gay, none of them have.
It's none of them, because one portion
It's
is
none of
darker
them
than
go
the
down
key.
the whole list, you can see which is darker than that. Oh, yeah.
It's none of them.
All right, so now you are seeing a mixture of things. So what's happening is this is what we recovered from the victim, but it was also the victim. So you might see some banding patterns that match the victim, right? Because we recovered it from the victim, but you might also see some from the accused because that is the semen, okay? All right, looking at the next one. So we got a blood stain. We got Bob, Sue, John, and Lisa.
Of course, John always.
Okay, John. Now, here's another one. So this looks kind of like our two examples ago. So because we get half of our DNA from our mom and half of our DNA from our dad, our DNA fingerprint is going to contain the bands from mom and dad, okay? So if this is us in the middle, the child, right? And this is the mother, and this is the father, we're gonna have half from mom and half from dad. So you're going to see some from both, okay? Similar to 2 slides ago. So you're gonna see some from the mom and some from the dad. So this is showing us 2 possible fathers, but obviously the one in blue, these are the ones that match up with the child. So you've got dad, mom, mom, dad, mom, dad. So a lot of times when you're looking at one of these, you're gonna see half from mom and half from dad. Okay, so in a small one like this, you see 3 from each. All right, here's another one. We got Mary, Bob, Larry, and the child. So here's our band from the mom, matches the child, right? So who is the dad? Larry. All right, here's a good one. So you got mom and dad, and you have child 1, 2, 3, and 4.
When to be child one.
They're all the children. Child
When to be
Yeah,
child one.
one.
1 and what?
3.
1 and 3.
Yeah.
Okay, this one gives it away because of the colors matching up,
Yeah.
but 1 and 3, if the colors weren't there, you have to match up these bands, okay? All right, so let's take a peek at this one. Assuming C is a parent of B, who is the other parent? So C is the parent of B. So who is the other parent? A1, A2, or A3?
A2. No, wait. Yeah, A2. B?
A3.
It's A3.
No, A1.
It's A, wait.
A2.
A2.
think
A2. It's A1.
it's A1.
It's A1. It's A, A2, got it, A2.
C, C is the parent of B.
Yeah, so it's
Therefore,
A2.
Say C is the mom. All right, just say C is the mom.
It's saying, yeah.
A2, A2, yeah.
Okay.
I know, I misread it.
Okay, so the father is
Right here.
A2, right?
So assuming C is the parent of B.
A2, yeah.
So this is the child, right? So these bands have got to match up. So this has got this one, and this one, and this one. So who has this
A2.
one? Okay,
I'm proud of myself, I actually got
nice job.
it every now and then.
All right, so we got mom and the child. Could this be the father?
Yes.
Yeah.
But they match up.
Okay, so mom.
Yeah.
Dad,
No one.
mom.
Yeah, it could be.
Okay, so it could be the father.
Could they match up and not be a?
You might be the,
Not typically.
really good DNA test, you might be.
It's a father. Where's
Where's the alleged?
So which children or child were fathered by the alleged father? So this is the alleged father.
Child two.
Okay, any questions?
I don't believe so.